First of all, I would like to thank for the opportunity to participate in this year's Biotech Atelier forum. In my talk, I'll try to figure out the current state of the molecular diagnostics of epilepsy and to feature some perspectives. Epilepsy is one of the most common neurological diseases affecting more than 50 million people worldwide. Their health, psychological functions, social life, and occupations. It is important to know that up to 70% of epilepsy patients could live seizure-free if properly diagnosed and treated. For this purpose, we need to understand the clinical and genetic characteristics of epilepsy. In only one-fourth of the incidences, there is an evident underlying acquired cause. Our understanding about the etiology of epilepsy have changed significantly during the genomic era, and now we know that genetics plays a major role in the remaining three-fourths of the epilepsy cases and could even contribute to the phenotype expression of certain acquired forms. The role of the genetic testings is to bring the precision medicine to the patient. It may lead to accurate earliest diagnosis, having a great impact on the disease management, the proper treatment plus selection, and the genetic counseling, allowing the prophylaxis of the subsequent pregnancies. Epilepsy displays extreme genetic heterogeneity even among patients in the same family. All known traits of inheritance are observed, and 60 to 80% of incomplete penetrance in different forms of epilepsy. Phenomena as genomic imprinting and uniparallel disomy in Angelman-Prader-Willi syndrome and a unique X-linked inheritance in sporadic infantile epileptic encephalopathy caused by mutations in the protocadherin 9 gene are also available. The molecular pathology encompasses germline and somatic mutations of various sizes, including chromosome aberration, exonic indels, and point mutations. Recent advances in the sequencing technology have led to the identification of the causative variants in many patients, fueled the novel gene discoveries, and changed our understanding about the role of different variants in the molecular pathology of epilepsy. For example, de novo variations that are the major cause of epileptic encephalopathies resulted in the identification of many additional epilepsy genes. Two major developments have fueled recently the studies of common variants. The first one is the increase in sample size made possible by the collaborative efforts of the international epilepsy research community. It led to the identification of 16 statistically significant risk alleles in the largest epilepsy study of Euroepinomics project. The second one is the use of polygenic risk analysis, which highlighted that common variants cumulatively explain approximately one-fourth of epilepsy risk and as much as one-third in the generalized epilepsy. Entering the postgenomic era, the paradigm shift brought new insights to the genetic architecture of epilepsy and have already revealed new genetic mechanisms and complexities of genotype-phenotype correlation. The most recent study that have been published in April reveals the potential complex genetic basis involving non-coding genes and double CNVs in neurodevelopmental disorder patients presented with epilepsy. The genetic and phenotypic heterogeneity of the epilepsies favored a dynamic diagnostic approach driven by the clinical data. There is no single test that could catch the disease-causing variant. In the genetic lab, we use different genetic technologies to look for variants of different types. Each test has its own advantages and limitations, and the major challenge remains the interpretation of the genomic data. To pick the disease-causing variant among the vast amount of background genetic variations is a long-way process. The average human exome contains many thousands of variants, including missense variants, protein truncating, small indels, and large structural variants spanning multiple genes. Identification of pathogenic variant requires rigorous evaluation of multiple lines of evidence like population frequency, protein localization, and evolutionary conservation. Criteria have been developed for determining the pathogenicity of variants for gene discovery, gene curation, and clinical diagnostic purposes. The prior identification of variant in a patient with epilepsy does not establish its pathogenicity, as some published epilepsy variants have been later reclassified as benign. During the last 10 years, at the Molecular Medicine Center and the Laboratory of Genomic Diagnostics, we have developed the following analytical approach for molecular diagnostics of epilepsy and epilepsy-related syndromes. It applies a single gene analysis in the beginning by Sanger sequencing and MLP analysis for all epilepsy patients, and this gene is SCN1A. If we have clinical data for hypoglycemia, SLC2A1 gene is also screened, and KCNQ2 gene in the case of benign neonatal infantile forms. The negative finding is a criteria to go for target gene panel sequencing and analysis of epilepsy-related gene panels. In epilepsy plus syndromes, this analysis is preceded by array CGH, since in 15 to 20% of cases of epilepsy plus syndromes, copy number variants being found as pathogenic disease-causing variants. Recently, the whole exome sequencing have been implemented for diagnostic purposes in our lab, where we perform analysis of epilepsy-related gene panel, but also the non-targeting variant analysis. Variant classification is based on the criteria of American College of Medical Genetics, and the variant evaluation includes also downstream genetic and in silico analysis to test for potential pathogenic variants. A very important part of this work have always been the sample and data collection process that had started many years ago and recently have resulted in the establishment of the Biobank at the Molecular Medicine Center, Medical University Sofia. It was recognized as National Biobank Hub in 2018. Epilepsy Biobank include DNA samples from more than 600 epilepsy patients and their relatives collected through research projects or diagnostic process. Extensive field studies conducted in large consanguinity families allowed mapping of novel genetic loci for epilepsy. Participation in collaborative studies, University of Antwerp and University of Australia led us to contribution to multi-center studies of Epicure Consortium. Recently, new research projects are launched, resulting in successful implementation of modern genetic technologies in the molecular diagnostics of epilepsy. Thank you for your attention.